Nephrons' functions and control. Nephrons serve as the operational units within the kidney, undertaking the roles of blood purification from harmful substances and maintaining a balanced blood composition in the circulation. This equilibrium is upheld through processes of filtration, reabsorption, and secretion. Nephrons also play vital roles in regulating blood pressure by producing renin, the generation of red blood cells through erythropoietin, and enabling calcium absorption via the conversion of vitamin D into its active form. Nephron structure and tubules. Each nephron comprises a dual-component system, a dedicated blood supply and an intricate network of ducts referred to as tubules. Within this configuration, an incoming afferent arteriole supplies the glomerulus, a specialized capillary bed characterized by elevated pressure. The glomerulus filters blood, producing a fluid known as filtrate that is subsequently collected by the nephron tubule. This tubule's proximal end is enclosed by the Bowman's capsule, forming an interconnected structure called the renal corpuscle. The filtrate, or captured fluid, travels sequentially through the nephron's tubule segments, the proximal convoluted tubule, loop of Henle, and distal convoluted tubule, before exiting the nephron through shared collecting ducts. While all nephron glomeruli reside within the cortex, certain nephrons have shorter Henle loops, classified as cortical nephrons. Approximately 15% of nephrons exhibit elongated Henle loops that delve deep into the medulla, named juxtamedullary nephrons. Blood circulation and capillary networks. After glomerular filtration, blood departs via the efferent arteriole, branching into a secondary capillary network encircling the tubule, recognized as peritubular capillaries. In the case of juxtamedullary nephrons, a specialized segment of the capillary, referred to as the vasa recta, extends deep into the medulla, following the loop of Henle. As the glomerular filtrate advances through the tubule, these capillary networks efficiently recover a substantial portion of solutes and water, redirecting them back into the circulatory system. This arrangement, characterized by a sequential capillary setup, echoes the concept of a portal system observed in other biological contexts, such as the hypothalamus-pituitary axis and the hepatic digestive segment. Filtration mechanism and selective barrier. The process of blood filtration is carried out by the glomerulus, while the peritubular capillary system undertakes the retrieval of various substances from the tubule. The efferent arteriole facilitates the connection between the glomerulus and the peritubular capillaries. The renal corpuscle consists of two key components, the glomerulus and the glomerular capsule. The fenestrate capillary structure of the glomerulus, marked by sizable fenestrations, is interposed between endothelial cells. Capturing the resultant filtrate is the role of the glomerular capsule, channeling it toward the PCT. The outermost layer of the glomerular capsule is comprised of simple squamous epithelium, transitioning to specialized podocytes that envelop the glomerulus. These podocytes are equipped with finger-like extensions known as pedicels, forming filtration slits that enable the passage of substances below a certain size through sieve-like gaps. This mechanism facilitates the passage of around 10 to 20% of plasma through the filtration process, contributing to the formation of the filtration membrane. Filtration membrane and regulation. The filtration membrane functions as a selective barrier, impeding the passage of blood cells, large proteins, and a majority of negatively charged particles. Substances with smaller molecular sizes, generally under 4 nanometers and often up to 8 nanometers, can traverse this barrier freely. Negatively charged particles encounter resistance due to the negatively charged proteins present in the membrane. This characteristic hinders the passage of negatively charged particles while allowing positively charged substances to pass through with greater ease. Additionally, mesangial cells within the filtration membrane contribute to the regulation of the filtration rate. This intricate mechanism results in the creation of a filtrate devoid of cells and large proteins, enriched with positively charged components. Proximal convoluted tubule, also known as PCT. Following its collection by Bowman's capsule, the filtered fluid enters the proximal convoluted tubule. This tubule is lined with cuboidal cells featuring abundant microvilli on the luminal surface, forming what is known as a brush border. These microvilli serve to amplify the surface area of the tubule, optimizing the absorption and secretion of essential solutes such as sodium ions, chloride ions, and glucose. Given the active transport of ions across these cell membranes, mitochondria are prevalent to generate the necessary ATP. Loop of Henle. The loop of Henle comprises both descending and ascending segments, situated near each other after making a hairpin turn at the lowest point of their descent. The components of these segments differ. The descending portion consists of both short and thick segments, while the ascending segment begins with a short and thin section, followed by a longer and thicker one. The composition of epithelial cells varies throughout the loop. The thick ascending segment is reminiscent of the DCT. Distal convoluted tubule, also known as DCT. The distal convoluted tubule, like the PCT, consists of simple cuboidal epithelium, but is shorter in length. These DCT cells exhibit fewer microvilli on their apical surface and display reduced activity compared to their PCT counterparts. Despite this, the DCT is responsible for transporting ions against their concentration gradient. Consequently, it houses a significant number of mitochondria, though fewer compared to the PCT. Collecting ducts and water regulation. Although distinct from the nephron, collecting ducts play a pivotal role in the final adjustment of the filtrate. Each duct gathers filtrate from multiple nephrons to refine its composition. As these ducts descend deeper into the medulla, they merge to form approximately 30 terminal ducts, which then empty at a papilla. The simple cuboidal epithelium lining these ducts facilitates the efficient transport of water. Juxtaglomerular apparatus, also known as JGA, and renin production. Adjacent to Bowman's capsule and the glomerulus, the juxtaglomerular apparatus plays a significant role. At the point where arterioles enter and exit Bowman's capsule, the initial part of the distal convoluted tubule comes into direct contact with the arterioles. This section of the DCT is referred to as the macula densa, consisting of cuboidal epithelial cells responsible for monitoring the fluid's composition. These cells release signals such as ATP and adenosine in response to changes in sodium ion concentration. Additionally, they house a non-motile cilium that gauges fluid movement within the tubule. The signals released by these cells help regulate blood flow and filtration rates. Furthermore, macula densa cells regulate the release of renin from juxtaglomerular cells of the afferent arteriole. Renin's role in initiating the production of angiotensin II, a potent vasoconstrictor, and aldosterone, a hormone affecting blood pressure, is crucial. JG cells and blood flow regulation. The juxtaglomerular cell, also known as the granular cell, is another integral component of the JGA. These modified smooth muscle cells line the afferent arteriole and possess the ability to contract or relax in response to ATP or adenosine signals released by the macula densa. By influencing the contraction and relaxation of these cells, blood flow to the glomerulus can be regulated. Moreover, JG cells produce renin, initiating a cascade of events that profoundly impact systemic blood pressure. Nephron summary and fluid handling. The nephron, the functional unit of the kidney, encompasses the renal corpuscle, PCT, loop of Henle, and DCT. While cortical nephrons feature shorter Henle loops, juxtamedullary nephrons boast extended loops reaching into the medulla. The glomerulus acts as a sieve, filtering blood based on particle size. The resultant filtrate is captured by Bowman's capsule and directed toward the PCT. The filtration membrane, a collaborative structure of podocytes and capillary endothelial cells, regulates filtration rate. Specialized cells within the JGA release signals to regulate blood flow and filtration, while others produce renin to manage blood pressure. The PCT handles crucial absorption and secretion processes, while the loop of Henle's distinct segments governs solute and water permeability. The DCT, although less active than the PCT, also contributes to ion transport. Collecting ducts, although not strictly part of the nephron, further refine filtrate composition.